Welcome, as always, everyone, for our Wednesday show with Dr. Kelly Victory. Obviously, we've been away for a couple of weeks, and Dr. Kelly has been holding the fort down quite nicely. But today we are joined by Dr. Ryan Cole. He is a pathologist. Let me get the exact specifics for you. He was trained at Mayo Clinic. He did a subspecialty fellowship in dermatopathology. He also has a PhD in immunology and virology, currently CEO of Cole Diagnostics, an independent full-service medical laboratory organization. Dr. Cole has some interesting ideas, and we thought we would hear them out. And Dr. Kelly has been talking about the kinds of things he's been observing for a long time. We also are out on Twitter Spaces, and of course, usually on Wednesday Kelly and I are pretty busy talking to our guests. We may not have time to take any calls. But if we do, and you raise your hand, I bring you up, you'll be streaming on multiple platforms. We're also watching you on Rumble and the Rumble Rants, and of course on the Restream, we get to see all the chat going on there, so I'll keep an eye on that. But let's get right on with it.
Our laws as it pertains to substances are draconian and bizarre. The psychopaths start this way. He was an alcoholic because of social media and pornography, PTSD, love addiction, fentanyl and heroin, ridiculous. I'm a doctor, for Christ's sake. Where the hell do you think I learned that? I'm just saying, you go to treatment before you kill people. I am a clinician. I observe things about these chemicals. Let's just deal with what's real. We used to get these calls on Loveline all the time. Educate adolescents and to prevent and to treat. If you have trouble you can't stop and you want help stop it, I can help. I got a lot to say. I got a lot more to say.
As always, Dr. Kelly Victory will be in here with my guest and myself in a few minutes. Susan, everything good with you?
Just perfect.
Just perfect. All right. We are back from our trip. I don't think you've spoken to this audience. We did a couple of earlier shows in the week, taking phone calls and whatnot, but we had a glorious time. But it's great to be back with you all. So let me get right to my guest. As I said, Dr. Ryan Cole, a pathologist, dermatopathologist, also immunology and virology training. Dr. Cole, welcome to the program.
With you. Thank you so much.
Oh, no, we don't have. There you are.
There I am.
Your sound was out for a second. Yeah, your sound was gone for a second. We got you back. All right.
I probably chimed in a second too early. Great to be with you. Thank you. I've actually listened to you for many years. When I was in med school, I was building wooden canoe after, post-call third and fourth year and was listening to Loveline. So it's an honor to talk with you. I want to just throw real quick before I get any more grief. I don't have a PhD. I did PhD work, and then we had our first baby on the way. And so I didn't finish the PhD. I didn't need to be a doctor. I had to feed the kids instead. So have my MD,
there you go.
Did PhD work. And so just for the record, thanks.
I'm glad you said it straight. It's perfect. But in terms of your post-call nights listening to Loveline, the only strange part is the wood canoe part of the story. What were you doing? What? Where were you? Were you up in northern Canada or something and trying to breaking the ice to go? What was happening?
In Minnesota, I took that wood canoe up to those north woods many a time. But it was kind of, I call it sawdust therapy. Post-call, you've got to do something creative and unwind. And so it was, I love doing wood art and creating. And so that life and death by day and a little art to balance life by night.
Yes, I think we all understand what you're talking about. So speaking of life and death, let's talk a little bit about, I'm not sure where we should start this conversation. I think what I'd like to ask is, there's sort of a couple ways to proceed here. One is the excesses and the extraordinary incompetence and missteps on the behalf of our public health organization and the extraordinary authority they were granted with fiat control over the entire populace with no requirement to defend what they had done. We're now understanding sort of where some of this came from, and most of it, frankly, from the Chinese Communist Party, and it was a completely fallacious way to go about fighting a pandemic, but okay, mistakes were made. So we could go that way, or we could start to talk about the vaccines and your concerns about the vaccines. And if we start with the vaccines, I'd be curious to know what's on your mind today. And we'll work our way through all your concerns.
So I agree in terms of public health policy and philosophy and what's happened. It has been a public health disaster, unfortunately. And I do serve now on our Central District Health Board as a public health official. When I saw what was going wrong, I thought, wait a minute, I need to get involved. And went ahead and ran for that board position, and thankfully was appointed there. And I'm trying to have a more logical influence in the community. But going to the second question, I think my concern today is we're still seeing a big public push for an injection that's still essentially experimental and has side effects, and even technically more so is not designed for the variants that are becoming dominant. And it will unfortunately never be able to keep up. This is the one thing I did learn in my PhD work in virology. Coronaviruses always mutate. And if you look at the history of SARS-CoV-1, MERS-CoV-1, there was a good reason we never successfully developed a vaccine against those. And that was the nature of the family of viruses. And I like to compare this to HIV. Dr. Fauci's holy grail of vaccine, an HIV vaccine. Well, there's a reason 40 years later we still don't have an HIV vaccine. Similarly to coronaviruses, HIV has a spike protein of different proteins, but same basic construct. And over time, that continues to mutate ahead of any vaccinal efforts. So with what we are trying to do to keep up with variants is a biologic and evolutionarily insane construct. It just doesn't make sense to be doing what we're doing. And so that's my big concern right now. We're pushing something that has risks to it. And certainly there are claims that there are great benefits. However, do the risks outweigh the benefit? In which age categories? In my medical opinion, we've got to assess all of those things. And in a healthy, robust population, at essentially zero risk for the virus, we're pushing something that can cause more harm than good. And I know we'll go into some of the science details.
Yeah, well, let's go there. Let's go there, because first of all, it's just I have to ask the question: do you have any theory why? Kelly and I have spoke to a lot of different people, and the kind of predominant theory that comes down is that there's an excessively cozy relationship between the governmental regulatory agencies and big pharma. Not that there's somebody twirling their mustache with a big bag of cash. It's just that these people move from regulatory positions to executive positions at the drug companies, and it's just it's almost a shared culture, and they don't question each other maybe the way they should. Whatever it is, it feels like that might be the reason that things are proceeding the way they have. But I still, even with that being the case, don't understand why this is the only vaccine I can think of that doesn't have either risk populations or age constraints over generally speaking, less within the construct of the FDA. I mean, look at the HPV vaccine. I mean, it's, what is it now, 9 to 46 or something? But you could give it to a 75-year-old. I mean, you could do it. I understand that. But in terms of pushing it on a 75-year-old, that's an entirely different matter. It literally is the same as me pushing a yellow fever vaccine on you, even though you have no plans to travel to endemic areas. What do we think that is? Why that? It's so confusing.
Yeah, don't worry. Uncle Sam gave me that one when I was at the Air Force Academy many years ago. But so I did get the yellow fever shot, and it was miserable.
Yeah.
Why is that? That's a good question. I agree with you. All scientists agree when you censor the ones who don't. And that's the unfortunate aspect of this. There have been financial interests. There is a coziness between regulatory agencies and big pharmaceutical companies, and it was obviously a boondoggle for these companies. But going back to the basic science, even if this had been a traditional protein vaccine, the SARS-CoV-1 protein vaccine, it induced an immune response, antibody response, et cetera. However, down the road in studies, it became, let's say, an enemy to the immune system because the immune system was blinded to future variations. So this is what we call immune imprinting. And it basically said to the virus, okay, focus on this spike protein, focus on this. And when along comes another, you end up with immunologic problems. And we knew that in mammal models with other viruses. it was fascinating that Borla in the Washington Post interview, CEO of Pfizer, said to his scientists, why are we doing mRNA? We've only been working on this for 2.5 years. So he had questions, and his scientists just said, well, it's because we're doing this, and that's our plan. And it was also interesting that Moderna hadn't successfully brought an mRNA product to the human market prior to this. Their animal studies with different toxicities and different autoimmune effects, et cetera, with mRNA technologies didn't work either. So it is, that's the $1 million question. Why did we push forward with something? Why did we not age stratify? And why did we treat these products as though they were, quote, vaccines when they're indeed an injection of a gene sequence? And didn't go through the regulatory necessary processes to authorize something that's a gene-based product. I mean, that's a very strict criteria in the FDA. And I was on with Dr. Weissman last night on a call and a couple other colleagues around the country. And we were discussing this. If you look at the handful of gene-based products that are FDA approved, the rigor through which those things go for approval is 5, 6, 10 years, and then with very stringent follow-up requirements. So all of those rules and that regulatory process that should have happened was set aside. They didn't do gene mutation studies. They didn't do cancer cytotoxicity potential studies, reproductive toxicity studies, weight-based analyses, and then the long-term follow-up. We're in the middle of it right now. We're in the short term. The long term is yet to be seen. And I hope for the best, but as a scientist and observer of data and observer of patterns and observer of cells, I have to say what I'm seeing. And what I'm seeing are some adverse effects that are highly concerning, not only for now, but the immune modulation that we're seeing going forward has me concerned as well. So I don't mean to sound alarmist. I'm just a scientist. My job as a pathologist is to be the quality control of medicine. You know, some people think, oh, pathologist, Quincy, autopsy. Sure, I review autopsy tissues. But all day long, I'm looking through this microscope at pieces of people. I'm reviewing blood studies, et cetera. And just like, you were traveling for the holidays recently, you hear in the airports all the time, if you see something, say something. Well, that's the job of the pathologist, is observe patterns within their community, within their populations. And if something is different, be it an infectious disease outbreak when we're seeing something new grow in the microbiology lab, or if we're seeing an outbreak of a certain type of cancer, et cetera, these things need to be mentioned and reported, because now you test the hypothesis against other regions and other labs, and you bring it to the public health agencies and say, we've got a change in pattern. And so in pathology, that's what we are supposed to do. And so obviously I've been lambasted for trying to share science. And at the end of the day, I always say to my colleagues, look, anything I do share or say, question me, please. Let's have dialogue. And if you have better data sets than I do, great. I'm here to learn as well. I'm not here to expound, ex cathedra pronouncements that what I say is the only thing. But when I do see something and it's concerning, that's why I'm pointing out the harms of the spike protein, the harms of these gene-based injections. The lipid nanoparticle in and of itself is toxic. And here's probably even a graver concern I have. So we've experimentally used a lipid nanoparticle plus a gene sequence for these injections. And the Wuhan spike that is coded for within this gene that our cells are taught to make this foreign protein. The human body's cells weren't made to make foreign proteins. That sequence is extinct in humanity. It went extinct over 1.5 years ago. The BA4 and the BA5 in the bi/trivalent shots is now also on the wane and almost extinct. So we have extinct variants, and the lipid nanoparticle goes anywhere and everywhere in the body. It was originally designed for carrying chemotherapeutic agents across the blood-brain barrier and or carrying genes for rare genetic conditions. So the lipid nanoparticle going everywhere is a dangerous construct in the sense you can't control where it goes. So now the companies say, well, we have an RSV and an influenza shot in development, and HIV and TB and 15, 20, 30 others in the pipeline. And they're acting as though they have regulatory carte blanche to move forward with a technology with known risks and harms, excess harms that I know Ed Dowd brought up some of the concerning statistical data with you the other week. with known harms, and now they think they can use this platform, and it hasn't gone through the proper regulatory processes. And that's what's super alarming for me right now. Okay, sure, we've experimented with these injections, but they're acting as though they have permission to go forward with other injections. And again, the human cell is made to make human proteins. To make a human cell make a protein from an infectious agent, and especially make a protein which has known cytotoxicity, cellular toxicity, human body toxicity, that's a big scientific concern.
Are there other pathologists that are, now that it seems like some of the silencing of our peers has loosened a little bit and people are able to say things out loud that they couldn't say a year ago, are you finding other like-minded pathologists?
Not as much as I would like to. It's similar to a lot of medicine and a lot of specialties. There's about 10% who are saying, hey, we don't think this shot is for everybody. There's probably about 25 to 30% that are like, okay, we're seeing the problems. A lot of people are, I think, in fear because they're stuck within large systems. Other pathologists around the world, yes, I have colleagues in Germany. Some of the slides I will share are from one of my colleagues there, from meeting with him when I was over in Vienna a couple months ago. And here stateside, yeah, I do have many colleagues that have approached me at meetings that are pathologists and say, hey, thanks for speaking up. I'm seeing what you're seeing, but if I say it, I will lose my job. I'm independent and I've lost just about everything in terms of my finances, career, business for simply sharing science.
Well, I assume the slides are, when you say the things you're observing, you're going to tell us that in the slides you brought us. So why don't we take a little break and we'll let you show the findings and then give Kelly a crack at you. So we'll be right back after this.
Thanks.
I have some pretty exciting news. Our favorite skincare brand, Genucel, is having a holiday preview sale. It just went live for all the products that Susan and I love. Genucel's Silky Smooth XV Moisturizer soaks right into my skin instantly, and with its immediate effects, you can see the fine lines and wrinkles disappearing within 12 hours. And Susan loves, of course, the Genucel Vitamin C Serum, infused with the purest vitamin C that absorbs to the deepest layers of the skin because of their proprietary skincare technology.
I am a snob when it comes to using products on my face. The dermatologist makes a ton of money from me. But when I was introduced to Genucel, I was so happy because it's so affordable and it works great. I was introduced to the Ultra Retinol Cream, which I love at night. All the eye creams are amazing. People notice my skin all the time, and I'm so excited because it's actually working.
And for a limited time, take advantage of the Genucel Holiday Preview Sale and save up to 60% off our favorite Genucel products. 60% off. Treat yourself this holiday season. Go to genucel.com/Drew. That's genucel.com/Drew, genucel.com/Drew. My guest is Philip Patrick. He is a precious metal specialist, trains at University of Redlands. He has spent years as a wealth manager at Citigroup, and his current position is with Birch Gold Group. So gold has always been somewhat of a safe haven, particularly in times of great turmoil, much like our present moment, I imagine.
Gold has always traditionally been a safe haven asset. Gold specifically has always been about wealth preservation, right? Gold has always held its buying power. You can look at as far back as you'd like in history. In biblical times, 1 ounce of gold would buy somebody 400 loaves of bread. And today it does the same thing. So it's a store of value. But I would say in times like this, as you mentioned, it's particularly important when you're dealing with things like 40-year high inflation, the air that's coming out of a stock market bubble. These times in particular tend to drive gold and silver up quite significantly. If things are different, the solution needs to be different as well. So I encourage everyone to get informed, and we have a lot of good information here to help your listeners.
Just a reminder, I am not a financial advisor, and I do not give out financial advice nor investing advice. Birch Gold has an A+ rating with the Better Business Bureau, countless 5-star reviews, and thousands of satisfied customers. Check them out now. Visit birchgold.com/Drew and secure your future with gold. Do it now. Some platforms have banned the discussion of controversial topics. If this episode ends here, the rest of the show is available at drdrew.tv.
There's nothing in medicine that doesn't boil down to a risk-benefit calculation. It is the mandate public health to consider the impact of any particular mitigation scheme on the entire population. This is uncharted territory, Drew.
And of course, welcome, Dr. Kelly Victory. When I hear those words now, I think about what's going on in China, and I just that's the furtherance of the policy that we were establishing here in this country. How do you all like it now? Anyway,
exactly.
So, Kelly,
can I say something really quick?
Yeah.
Also, we're trying to reinstate Dr. Kelly on Twitter.
That's true.
And we made an appeal to Elon Musk today, and we're getting some traction over there. So go over to Dr. Drew's Twitter and retweet and tell a friend.
And tag Musk.
Yeah.
Right.
Please,
all right, Dr. Kelly Victory, have at it.
I appreciate it. Welcome, Ryan, and thank you so much for joining us. I've really been looking forward to this conversation. I think it's fair to say that COVID is what threw the two of us into the same circle. For the better part of the past 2 years, you and I have been immersed in the same group of scientists and singing really largely from the same song sheet, truth-telling, and unfortunately, both of us have suffered the slings and arrows as a result of it. But I applaud you for your honesty and your courage. I want to get into a little bit. You were the first person who I knew when we were talking about potential adverse events as a result of the vaccines. People started talking, the very first thing, well before people were talking myocarditis or autoimmune issues, it was the clotting issue. Clots were the first thing that people talked about. And you were the first person that I knew who was taking this from the clinical level of we're seeing people with heart attacks, strokes, blood clots to the lung, blood clots to the extremity. And because of your really unique purview as a pathologist and the one actually looking at the slides, looking at the autopsies or whatever, were the person who got into the weeds on these clots. Let's start, and I'm going to lead you through in my 45 minutes that I've got with you, a number of different sort of categories of adverse events that potentially are associated with these vaccines. But let's start with clots and go from there. Talk a little bit about the clots, what you are seeing, what they're made of, and whatever else you want to cover.
Thank you, Kelly, and it's an honor to be with you, and thank you for all you do. We can start with the clots and the spike protein in and of itself, and I think it's fair to mention, look, COVID itself was a clotting disease. And so what part of COVID was causing that clotting? The spike protein. And sorry, I'm bouncing a bit there. I'm just getting my slide up. So the spike protein, interestingly, and I will give Dr. Pretorius in South Africa a lot of credit for her work in terms of some of the mechanisms of clotting and the composition of these clots. So here, this is what the body's doing. We're taking a gene out of the virus, putting it into the spike, or putting that gene, into a lipid nanoparticle, putting that into the vial, putting it into the arm, and then putting it into our bodies. And then our bodies become a spike protein factory, and supposedly, we are supposed to make antibodies and immune response. So we'll look at the next slide here. If Caleb wants to jump one slide forward. So here you see a superimposition of a needle on top of a muscle. And you see in the middle that little circle, that's a vessel. And we were told, you get a shot, it stays in your arm, you're fine. But you see below, that's a little torn apart vessel. And then on the right-hand side, this is a technique we use in the laboratory called immunohistochemistry. So, people can say, well, gosh, why isn't everybody doing this? Well, that's my question, because here's a list of countless vendors of this stain, this ability to do this testing in any and every laboratory in the world. And why aren't my colleagues doing it? Where's the billions of dollars for research at the universities looking at every sudden death, every clot, every unusual scenario with both COVID, post-vaccine, et cetera? The billions have gone to the NFL, to other people to advertise, to get a shot, but the research should be done. So here in this picture on the right, all those brown dots, that's this technique. We can tag specific proteins in the body and in biology. So those brown dots are in the muscle, and those muscle cells are making spike protein. However, let's go forward a slide. You hear a lot about myocarditis. On the left, that's normal heart muscle. On the right, all those blue dots, that's inflammation that doesn't belong in the heart. Let's go to the next slide and see why. And I'll get to these clots. Don't worry, Kelly. I'm getting there. I'm just building up to it.
No worries. No worries.
I'll take 5 minutes and go through this, and then you can just pick my brain. So on the left here, that's heart muscle tissue. And see all that brown? That is all spike protein. The lipid nanoparticle, as I was mentioning to Dr. Drew, goes everywhere. It goes everywhere. It can land anywhere in the human body, any organ, bone marrow, spleen, brain, heart, et cetera, et cetera. So what do we do as a control? People will criticize and say, well, how can you prove that's not the virus? Well, on the right-hand side, the virus has lots of proteins, and the nucleocapsid that encases the mRNA is a specific protein. We stain for that as well as a control. If it were a viral infection, we would see both of those stains positive. Post-vaccinally, we see only the spike protein. So this is heart muscle. That's highly concerning. Does a foreign virus belong in heart muscle? No. Does the toxic protein from an injection our cells are making belong in our heart muscle? No. So sudden deaths, here's one explanation. Let's go on to the next slide. I know many of us have heard about Dr. Malhotra now, a brilliant cardiologist from the United Kingdom. I've had the pleasure of meeting him. His father, unfortunately, passed away after one of these injections. He had a clean bill of cardiac health, and then after the shot, ended up with cardiac blockage, cardiac vessel blockage. Well, here, the red, this is what I look at all day, different slides like this. All the blue dots in that red, that's hemorrhage and inflammation in a coronary artery vessel wall. The paler background, what you see there is spike protein stain, again in the laboratory, showing that these vessel walls are packed with production of spike protein. And a concern I have is, when does the body, after this gene goes into our cells, when does the body stop making the spike protein? That is a scientific question that hasn't been answered yet. Let's go on to the next one, please. Okay, here, this is a recent report by Dr. Mortz out of Germany, a case report. And this is to the point that lipid nanoparticles were designed to carry agents to the neural tissues. The blood-brain barrier is very sacred, and it prevents many things from getting into it to keep our neurons firing and safe. Well, this Parkinson's patient, deceased, you can see, again, all those dots on that little tube on the left, that's a blood vessel. That's all spike protein. And on the right, you can see spilling out from a vessel all the little dots, that spike protein within the neural tissues. Next one, please. And in this next one, again, this is another autopsy case, and this is spike protein just dotted and spread throughout the neural tissues. It induces inflammation. And wherever it lands, it's going to induce.
Can I ask a couple quick questions? I don't want to interrupt too much here, but where exactly is that? Is that in myelin being produced, or is that in neurons? What are we looking at? For me as a non-pathologist to localize that.
Yeah, no, it's in white matter and gray matter. So wherever the capillary vessel is, the spike protein.
Okay, is it? So let me make sure I get your theory correct. So it's primarily being reproduced in endothelium, and then leaking out into the surrounding tissue. Is that correct? And so it's really why is the endothelium so prone to the production of the spike protein? Do we know that?
Well, the endothelium is replete in ACE2 receptors. The production site. So the spike will preferentially bind both to the ACE2 receptor on the endothelium, but there's another very important one, and this is what we'll talk about in the clotting, called the CD147 receptor. And that protein we have on the surface of red cells, white cells, endothelium, platelets, and that spike preferentially binds to that. Now, I won't tell you about a drug that shall not be named that displaces that spike from that receptor, but there are medications that do displace the spike from those. But
and let me just, before you keep going, one last quickie, which is how does we're looking at post-vaccine cases. If we looked at post-COVID cases or even active COVID cases, is there any similarity?
Yes, there is. So NIH did one of their rare studies on a series of autopsies, and patients that were immune suppressed were replicating virus, and they did both that spike and nucleocapsid stain, and they showed that the virus did get into many parts of the body and replicated for a good period of time. So there are comparators to post-COVID deaths and autopsies compared to post-vaccinal.
Right. Okay. But if we summarize so far, and I'm going to, part of my job is to make sure that we don't get so far into the technical weeds to lose those folks who don't have science backgrounds. But so far we said we knew that COVID was associated with this spike protein that by itself is toxic. It is what caused clots in those people who contracted COVID and had clots. They develop a vaccine that induces you to produce those precise toxic spike proteins, and with no off switch. Unlike when you get the virus and you will eliminate it with the vaccine, you now have the genetic roadmap or instruction manual to make these in perpetuity, perhaps. We don't know for how long, because those studies were never done. So when you get vaccinated, you now are going to produce the toxic spikes. And furthermore, they're going to be produced in essentially every organ system in the body, despite what we were told. They won't stay in the deltoid. Furthermore, because of the nature of the stains that you used that were available, you were able to determine the difference between spike proteins that occurred from the virus versus spike proteins that occurred from the vaccine. And to differentiate that, okay. So far, you now have become, if you're vaccinated, you're now a little factory producing gobs of toxic proteins in every organ system in the body. Go from there.
And I want to clarify, to just take some fear away from some individuals. The vector with the J&J and the AstraZeneca is an adenoviral vector, and you're starting with complementary DNA, not an mRNA. And so because of that, you don't get the breakdown. It produces a high amount of spike for a set period of time. We know from the 1980s, 90s that gene-based studies with adenoviral vectors caused a lot of clotting. So there were clotting problems with that one for sure, and some antibodies in the blood that also induce clotting, platelet factor 4, antiphospholipid antibodies, other things in the body that will induce clotting from that vector. But you don't get the replication for the same period of time with mRNA vaccines. Right. So
I had some sort of, I don't know if it was thrombotic thrombocytopenic purpura or what. I had all the signs of a transverse sinus thrombosis. I woke up with a raccoon's eye and was sick as hell.
I saw that.
And made it through. But that's what you're talking about. So there it is. There's my raccoon eye.
Yeah.
So
keep going,
now
Dr. Cole.
move us forward.
Yeah, we'll move forward real quickly here, and I won't be too nerdy. I'll try to be a little more layman's here. So if we jump onto there we go. So it's in the brain tissue. Dr. Drew had a great question. Is it in the gray matter? Is it in the white matter? Where in the brain? Wherever the lipid nanoparticle distributes, and it does get through the blood-brain barrier, and we know S1, the spike gets through as well. So it'll follow the small capillaries and leak into whatever tissue it wants to. Okay, next slide, please. Now, this is the aorta. This is something that's unusual. Usually you'll see this in genetic conditions where the aorta ruptures. And you can see on the top, that's the autopsy tissue. On the bottom, all those dots, that's inflammation. On the right, that's spike protein, literally causing the lymphocytes to chew a hole in the aorta. This is the biggest blood vessel in your body coming off your heart. When that ruptures, you're gone in minutes. So that's just another example of what deposited spike protein and the induced inflammation can do. So next, please. This is just an example of how prolific the spike can become once it lands in a tissue. So that big circle in the middle is an artery inside your spleen. And all those brown dots in all those splenic cells, that's a lot of spike protein. And so we know the spike can circulate in the body for a long time. Traditionally, in vaccine injury from other shots, they say most injury occurs within 2 weeks. However, with these gene-based injections, the spike is persisting for a long time, and it's produced in large quantity. Dr. Ogata at Harvard showed it was circulating for a month. Dr. Bonsall in the Journal of Immunology showed it was circulating in exosomes in the blood for up to 4 months. So we have spike protein persisting in the human body for a long time. Now, in some immune suppressed patients, virus persists for a long time as well. So I'm not throwing one or the other under the bus. I'm just saying this is a scientific observation. So going on to the next one. This is what's concerning. Okay, so this is a lymphoma, a cancer of the lymphoid cells within the stomach lining. Now let's look at the next slide. All of those B cells are stuffed with spike protein. The spike protein binds to a gene in our body that's called the guardian of the genome, the p53 tumor suppressor gene. We know from in silico studies that the spike will bind there. It also binds to a breast cancer gene known as BRCA and ovarian cancer gene known as BRCA. What does that mean? Well, I've made the observation that there's an uptick of some unusual cancers that I've been seeing. But this, I think, this should just at least open the door to other pathologists and universities to say, well, we should maybe go back and look at some of these unusual fast-spreading cancers in young patients that we normally don't see. Is there spike protein? Is it binding to that receptor? Why is the body doing things that we haven't seen on a population level before? So again, I'm just putting it out there. These are things that are scientific, have been observed. These are observations. These are scientific methods. Others need to continue to repeat these. Okay,
and let me interject here with regard, if I can interject for a second here with regard to these cancers, Dr. Cole, you are certainly not the only one seeing these. There's been a significant increase in new onset cancers, particularly in young people, and a resurgence of cancers that had been deemed in remission. There was just a letter, a very compelling letter written to the British Medical Journal by a well-known British oncologist named Angus Dalgleish, who wrote that he said, please stop these. We are seeing this massive uptick in colorectal cancers and in 44 other classes of cancers, particularly in young people. So we need some explanation. And while you and I cannot say with surety, as we sit here today, that these are a direct result of COVID vaccination, we certainly are obligated, I think, to have that robust, vigorous debate. And somebody sure as heck should be investigating these and saying what's causing it, because the things you are bringing up with regard to the binding of the spikes to these different receptors on certain well-known means. One of the great advances in cancer was our ability to define receptors and therefore have targeted therapy. A receptor for breast cancer, a receptor for colorectal cancer, a receptor, for ovarian cancer. And now we have spike proteins that are binding to specific receptors and allowing tumors to grow unimpeded.
And that's the important thing, is there's at least a dozen mechanisms that the spike can induce in those cancer pathways. And that's, my scientific question and concern, what I've been observing. T cell suppression, your immune system keeps cancer in check. All of us have some atypical cells right now. Right. Your immune system knocks them out. Interferon is critical for fighting off other viruses as well as for fighting off cancer, especially type 1 interferon. The spike protein suppresses that in our cells. The spike protein damages our mitochondria. It can get into the mitochondria and inhibit the breathing of your cell, the energy of your cell, the ATP. So every cell in your body has these little factories making energy for you all day long. The spike protein impedes that. Great papers by Dr. Clough and Abramovich. So, and those are the tip of the iceberg. And then certainly microclotting, can cause choking off of tissues, and cancers do like oxygen depleted environment. So and I could, I won't get into the weeds on it. That could be another long lecture. But you're right. It's all these things added up into one that it's all of a sudden with what's new. Well, there's a new product that's never been used on humanity before that has plenty of plausible mechanisms, plenty of proven mechanisms. And yet the scientific questioning, the endeavor to say, hey, there's a signal. We need to look into the signal. We need to fund this.
Well, just for me, out of curiosity, what is the tumors you've been seeing? What was it, like a Burkitt's or something? What have you been?
Well, no, interestingly, and there is an increase of Epstein-Barr virus, both after COVID and after, and Burkitt's is driven by Epstein-Barr virus. That's a B-cell lymphoma that tends to be driven by Epstein-Barr virus. And interestingly, and about half of the long haul patients and COVID vaccine injured patients, they do have reactivated Epstein-Barr virus. So I encourage clinicians, whenever they have that fatigued patient post-vaccine or post-COVID, check Epstein-Barr virus titers to see if it's reactivated. My first signal was in women's health. I saw an uptick in endometrial cancers. That was the one that first concerned me. And then I do a lot of skin pathology. And so melanoma in a lot younger patients. And what was fascinating, especially after shot 2 or 3, was the activity of growth of the tumor. So we measure how the cells are dividing and at what rate. And it was dividing at, these tumors were dividing at far higher rates. I've been doing this for 26 years. I've seen 500,000 patients in my career. You kind of know the patterns. And when you see something different, you're like, gosh, that is a lot of cell activity in this tumor compared to what I've been seeing. And it's in like Dr. Victory said, in younger cohorts. And that becomes a concern as well. So again, I'm not throwing anyone under the bus, but this is an industry that has something new. And all of these things are the kind of things we should have done long before a rollout of something experimental on a large population.
All right, so I keep throwing you off the blood clots. So back to the clots.
No, let's get to the clots. No. I'm more freeform like this. So, I mean, I can get on stage and do a presentation, but we can jump a couple slides forward. Let's look at those clots because, there's been some sensational videos. And unfortunately, I didn't have any editorial power and didn't know I was going to be in one. I'm like, oh, gosh, here we are. And so I can dispel some of the controversy if I can. So, yeah, let's go ahead and jump a slide or two, Caleb. Here we are. So people say, oh, these clots aren't real, that's all made up, whatever. People are confabulating. These are from living patients. So these patients, fortunately, are still with us. So I have some surgical colleagues around the country that have seen an increase in their patients, thankfully done the scans, found them, and extracted them. So these thick fibrous clots are in vivo as well. These are in living patients. The interesting tube of blood on the right, this was a patient from my colleague, Dr. Burkhardt in Germany, and she had 3 shots. Whenever it would get cold, her fingers would start to hurt, kind of a Raynaud's phenomenon. They took a tube of blood, set it on the counter, and little fibrils started to form. They spun it down, and the material within this spun down big nugget there is the same as what we're seeing in the clots. So we'll go a slide forward. And this is what it looks like. It looks like just this pink amorphous material. Well, that's fibrin, it's reticulin, it's a bunch of amino acids, but more importantly, there's a material that the body doesn't break down very easily called amyloid. And you may hear about amyloid in Alzheimer's disease and plaques in the neurons in the brain. That's what we call a beta-pleated sheet amyloid. There are different types of amyloid, but the problem is the body doesn't easily break down amyloid. Our body can break down fibrin and clots, and you'll form a clot after scraping your arm or knee or whatever, and eventually your body remodels that area. And because we have enzymes that say, hey, there's a clot, it's in the way, let's remold it, put some new vessels in, all done, good to go. However, this type of proteinaceous material doesn't get broken down easily by the body. I don't want to scare everybody and say, gosh, everybody that has, the shot is going to have this. No, the good news is most people are fine. However, from a scientific point of view, you can't find what you don't look for. So early on, the agencies and Fauci encourage people not to do autopsies. So you're not going to find if you're not looking. The morticians that started seeing these, they were getting, when a body comes in and they have to preserve it, they cannulate large vessels, they put their needles in large vessels, they started getting back pressure that they hadn't experienced before. And there are 1 or 2 that have spoken out, but I know of about another 50 that are seeing the same, but again, want to keep their jobs. So they don't say anything. But when we look at these clots under the microscope, this is what we see. We'll go another slide forward here. Now, this is interesting. So here's autopsy tissue. This is a thicker clot within a larger lung vessel. And all that brown in the middle, guess what's in the middle of that? Spike protein. And again, it's binding to those little receptors we talked about. And Dr. Pretorius did a brilliant study where, the one from Dr. Reza Pretorius, South Africa, where she showed that you can take the platelets out of the blood and put the spike protein in, and it will cause proteins to clump together in the absence of platelets. So this is really unusual in clotting mechanisms. I ran one of the busiest clotting clinics in the world at the Mayo Clinic for a long time, and just understanding all the factors that go into form a clot. To see something like this in a protein that will do this, in the absence of other things that are usually necessary for a clot is very unusual and, again, concerning. Do I want to alarm people? No, I don't want to alarm people. Do I want people to be aware? And do I want my colleagues around the world to study this? Absolutely. So we'll go on to the next one here. This is just an example of the postmortem clots. And yes, they're long and they're very fibrous. And that fibrous aspect of them, there's no, nano chips and wires and all this crazy stuff you hear on the internet. I think that's just all tinfoil scaremongering. But what there is unusual amounts of collected proteins. There are unusual combinations of proteins that make these very difficult for the body to dissolve. We'll go on to the next one.
Correct me if I'm wrong. Let me just, if we keep referring to these, or people have referred to them as blood clots, these, foot long or a yard long, quote, blood clots. You're talking about what's in there. Fibrin is, kind of amyloid. But if I'm correct, they also are devoid of those things that we normally associate with blood, meaning there isn't hemoglobin or potassium or iron, the normal components of blood. They have a very different texture. If you roll a blood clot around in your fingers, it essentially will dissolve. If you roll these around, they have a very different texture, but they are devoid of typical blood components, correct?
And let me pile on with a question on the heels of that. And are they the same in postmortem as in somebody who's you're finding these in a living person?
No, and I've done about 600 autopsies earlier in my career. Spent a month in a medical examiner's office as well. Postmortem clots tend to be like a thick red jelly. And like Kelly was saying, you can squeeze them, you can kind of roll them around and just break them apart. That's the uniqueness of this. These are like rubber bands. And I do kind of want to dispel a little bit of what you mentioned, Kelly, because they do house normal blood elements. So they do have the fibrin you would see in clot. You do have some of the normal clotting proteins. It will entrap red blood cells. It will entrap white blood cells. So those elements are within it. The unusual aspect is the rest of the composition of this long fibrous clot is that amyloid-like material with some mixed sugars, glycoproteins mixed in. So it's the difference of being used to seeing a normal clot, having that part of it there, but this addition to it that makes it, elastic and odd. And
can I ask another quick question on the heels of this? I've had this very distinct instinct all the way along as we've learned about this illness, that there's a significant endothelial pathology of some type, both with the vaccine reactions and with COVID. I've almost, all the microvascular stuff in the brain, I'm convinced is really something happening in the endothelium triggering clot. Is there anything about what you're describing with the vaccine that sort of either evidence for or theoretically in your own mind that fits with that theory?
I think that COVID itself and injury from the spike protein from the injections is one and the same pathology. I think you're absolutely right. I think the primary disease is a system-wide endotheliitis, and that means inflammation of the lining of the blood vessels. And there are so much binding to that ACE2, to that CD147 that we mentioned earlier, and then these induce inflammatory pathways, cytokine pathways, but also, and most critically in this aspect of the conversation, the clotting pathways, be it microclotting, the acute blindness and retinitis that we see, acute neurologic change and cognitive brain fog, either from the virus or post-vaccine, COVID fingers and toes before the shots ever came out. And we still see that clotting pattern in many post-vaccine injuries as well. So it's that microclotting that I think is the principal thing that we need to address in medicine. Now, there's one thing that I've been a big advocate for, and that's derived from fermented soy, nattokinase, which is an enzyme that breaks down fibrin. And if you look at, again, parts of the world like northern Japan, they don't die from heart disease and strokes, and they eat a lot of this fermented soy in their diet. And I'm not your doctor. I'm just saying an interesting observation is to look at this. It's known as a fibrinolytic enzyme that's naturally available, both in food and as a supplement. And I've had colleagues who've ended up with clots after long travels and put on Eliquis and other drugs. And I said, have you tried nattokinase? And a day or two later, gosh, their clot is dissolved. So there are enzymatic mechanisms to start breaking these clots down before all these other amyloid proteins can start to piggyback on and compound and get bigger and bigger and bigger. So I agree with Drew 1,000%. COVID is a clotting disease. COVID is an endothelial disease. Yes, there are all these other phenomenally bizarre things that the spike protein does, and I could give hours lectures. I won't. But yeah, you're right. That is where it all begins. I think you're 1,000% right.
Increased intake of nattokinase in Japan is one of the theories for why they have they enjoy a very, very low rate of both Parkinson's and Alzheimer's because of the amyloid component. So it's an interesting theory topic for yet another show. So back to the clots. I need to back to clots.
Okay, back to the clots. Yeah, the clot thickens. Here we go. So in this picture down on the right, you can see. So those are some slides from my lab, and you can see some of that more pale blue area down below there. That's more of the glycoproteins, the thicker pink bands. That's the fibrin and the amyloid. But up above, you can see parts of it that are speckled with blue. That's why I just wanted to point out there are normal blood elements trapped within these. So part of them look like a traditional clot, really the add-on that makes them look weird. So we'll jump ahead to the next one real quick. And this here's my biggest concern. So this is what we were talking about earlier. And to Dr. Drew's point of this spike protein causing inflammation chronically within vessels, my concern is the persistence of this spike protein. So this was a brilliant study by my colleague, Dr. Rolkin at Stanford in the journal Cell. And she took lymph node biopsy after the injection, and all the pink that you can see on that first left column, that's mRNA. So at week 1, there's still mRNA because of this synthetic pseudouridine that's substituted into the sequence instead of our natural uridine. Our body normally breaks down RNA in a couple of hours or a day or two, depending on what protein our body's trying to make for our normal bodily function. So what was advertised by the agencies is, oh, mRNA breaks down. This is safe. You don't need to worry about it. No problem. However, when you put a synthetic molecule in these little beads of pearls, these nucleotides, it doesn't break down. And this study proved it. And so at week 1, all the pink in that first column, mRNA still present in lymph nodes. At week 2, at week 3, at week 5, at week 8. And then the next column over in the lymph nodes, at week 1, certainly spike is being produced, but guess what? In that same sequence of weeks and weeks and weeks and weeks. Now you see less, that's a good sign. Is the amount going down? It appears so. And is this happening in everybody? No, but in a set percentage in this study set, their body was still making spike protein, but more importantly, that mRNA was still persisting within their lymph node cells 2 months later. Now, here's an important point to add on. This study stopped at 60 days so they could go to publication. What happened at 90 days and 120 days? Some of the autopsy series I've looked at with my colleagues in Germany show that spike protein being deposited in high amounts 120 plus days after the patient's last injection prior to their demise. So again, in science, that precautionary principle, what's the risk benefit? Are we doing something? Do we know the long-term outcomes? We need to be studying these things in science. And I will take all the slings and arrows any colleague wants to throw at me. I am an observer and a reporter. That's what a pathologist does. We observe, we report. And the cells don't lie. And I like to say that. The cells don't lie. What is there and being expressed is there and being expressed. I mean, this isn't brand new science in terms of identifying these things. We've been doing this for decades in the laboratory. And so when you look at these patterns, now this is informed consent. This is giving the public informed consent. A bit too late, but these are the kind of things that our agencies should have done beforehand and said, oh, by the way, if we give you this mRNA for at least 2 to 4 months, it's going to be in your body, and so is that spike protein. That would be informed consent. And by the way, it causes an inflammation of the blood vessels of the body, and it can cause an inflammation of the heart, and it can cause inflammation of the brain, and any and every organ in your body. So that would have been informed consent, because were these things known in some of these laboratories beforehand? Well, if you add up the history of lipid nanoparticles and look at the toxicity of that, they knew. If you look at the failure of mRNA products prior to this, they knew. There's a long history, and just putting the pieces of science together, I don't have to go on any tinfoil pathway of any sort. I can take all the published literature and say, here's how we knew it was a bad idea. Here's how we knew. Here's how we knew. Here's how we knew. This is how science is done. You make observations. You obviously hypothesize things as well. Test, try, either succeed or fail, go back to the bench and try it again. But these things were known well before these products were put together. And now we're seeing, unfortunately, and I'm not here to criticize, I'm here as an advocate for truth and science. And if you got one shot, I'm not criticizing you, don't get another. If you got two, don't get a third. If you got three, don't get a fourth. Because number one, the coronavirus of any sort will always mutate ahead of what we try to do. And number two, based on what we're seeing scientifically in the laboratory now, the risk benefit ratio is all on the risk side. So that's the science behind it. And again, we can go into the immune pathways of the immune system attacking self. That's another huge problem is autoimmune disease and the autoimmune antibodies that the body's forming against our myelin, against our platelets, against tissues in our body.
Right.
There's so many mechanisms of harm of this spike, but that's kind of that's clotting in a nutshell, and a couple other scary things, but happy to go down any other pathway, Kelly. Sorry, I get excited about nerdy science.
No worries. And I would submit to you, by the way, that not only did they not give informed consent, we were told falsehoods. We were told, number one, that the injections would stay in the deltoid muscle when they knew that that was not the case. We were told, number two, that it would be eliminated from your body very, very quickly. Still, as of today, it appears on the CDC website that the mRNA gets eliminated within a matter of days. They know that that's a falsehood, and they always did. And we were told that it couldn't be incorporated into your DNA. And we have some pretty compelling, at least in vitro studies, that show that it in fact can be reverse transcribed or is reverse transcribed into cells. I am not anti-vaccine. What I am is pro-safety and pro-data. And these vaccines were rolled out far too quickly with a paucity of safety data. And I'm with you, they should be pulled at this point. I think we've covered, at least at a high level, the issues about the clots and how these vaccines might be associated with causing myocarditis. You showed those slides of the heart muscle. And even we've talked about the potential relationship to increases in cancers because of the way the spikes bind to those different receptors. One thing I'd like to also ask, if you can see a connection. We know, and Drew and I have done shows reporting on the somewhat alarming decreases in birth rates that are being seen around the country and increases in fertility issues. Is there something, how in your mind, from a pathologist's purview, do you tie together what you're seeing under the microscope with what is happening in the reality with fertility and birth rates?
Well, if you go back to the biodistribution studies that were FOIA'd out of Japan, and Dr. Bridle, I was on a call with him last night, that lipid nanoparticle, like I said, goes anywhere and everywhere in the body. Well, it likes to hone to fatty tissues, and the ovary has a lot of lipid in the cells. In those studies, that lipid nanoparticle was concentrating in the ovary at higher rates than other parts of the body. When they stopped that distribution study, the inflection curve, when they stopped it at 48 hours, was still going up. And unfortunately, they stopped at 48 hours, sacrificed the mice, and then looked for the distribution. If you look at that study, they cheated on that a little bit too, because they combined the male data and the female data. The accumulation in the female was a lot higher in a lot of the organs than in the male. We know that in male sperm, the spike protein post-vaccine study out of Israel showed a decrease in sperm count as well as decrease in sperm motility for 6 months. In the female body, the fallopian tube, the ovaries, the eggs themselves, going back to Dr. Drew's question, are replete with ACE2 receptors. That spike protein will hone to that ACE2 receptor that your body is making post-vaccine. And wherever that spike lands, it is an inflammation-inducing protein. And so inflammation in the ovary can, number one, kill off eggs, and number two, inhibit the ability for that egg to be robust in terms of implantation, in terms of getting to full ovulation and or release. And we've seen, and I don't think it's just ovary. Here's my other concern, and it goes back to the brain question and where can this spike go? Where can this lipid nanoparticle go? Our endocrine system is feedback. And so we have hormones here in our gonads, and we have hormones in our brain. And they say, okay, release the follicle. Okay, start making stimulating a new egg to develop, back and forth. So if we have inhibition in neurons for normal hormonal cycles, and we also have inhibition within the ovary itself, where the spike protein is binding, where we know the lipid nanoparticle preferentially concentrates, then if you look at the drastic decrease in fertility, and I like to joke, hey, look, we should have more babies than ever. People were locked up. There's something they should have been doing. So,
Right.
so it is a concern. And the European data, they're down on average 10%. And if you look across the board, about 110,000 births that are down. Taiwan data, same thing. Sweden, Netherlands, so many countries.
Yeah, Taiwan, I think, is
plausible mechanism. Now, here's the other sad thing, and I'm going to grab this from under the desk. This is starting to arrive. These are placentas. These are placentas coming from obstetric colleagues around the country. These placentas are the wrong size for the gestational age. These placentas are calcified. These placentas have spike protein in them. These placentas have antibodies in them. These placentas have induced excess inflammation in them. This particular one is from a nurse, hospital, 8 months pregnant, required to get the jab to keep her job. Very shortly thereafter, unfortunately, intrauterine fetal demise. I know you guys talked to a couple other colleagues that went over that kind of data. So certainly the data paints a picture, but more importantly, the pathology mechanisms are already identified, and just more studies need to be done from these. So what I encourage is any medical colleague anywhere in the world, find your pathologist, say, here's the list of stains, start ordering them, start looking at these things in the tissue of the deceased, start looking at these in those fetuses that don't make it to full term, start looking at those placentas. Start taking your surgical specimens that are in unusual cases of multiply jabbed individuals with unusual conditions and start looking for what would be causing it. Obviously, I have an inkling based on what I've shared, but yes, I think you're right. There are highly mechanistic, plausible mechanisms, great literature on this already. And I like to quote Mark Twain often I don't want to throw my colleagues under the bus in medicine. But Mark Twain said, the man who does not read has no advantage over the man who cannot read. And I think this is the problem we're having with kind of this groupthink of safe and effective, no problems. My counter argument is, but look, read. It's here. I mean, it's hiding in plain sight. Don't just believe a narrative. Like Rochelle Walensky said, she was getting her vaccine safety information safety from CNN and just chuckled about it. I'm like, good grief. I mean, these are people responsible for countless lives. And going back to certain data points, those cancer signals, that's up to the CDC to be up to date and reporting those in real time. You and I are hearing these signals of all sorts of things as we go to meetings from colleagues. I'm hearing from oncologists, interventional radiologists, left and right, of seeing these things in 20, 30, 40-year-olds, and they're alarmed. They're like, we're seeing it. I got a call from an OB, I won't say in which state because she is, a tall blade of grass, but she said, is there a better system than VAERS? Because I am seeing leiomyosarcomas in the vaginal tract and in the uterus at rates I have never seen in my career.
Yeah.
And she's in a state that's highly jabbed. So these are all signals. We need to do the science. I'm a voice for scientific observation. Like I said, you can criticize me. That's fine. Bring better data, but do the science.
I have a clinical question.
Well,
which is seeing a lot of very rapid junctional rhythms in young males. I've been thinking that it was probably some unseen, subclinical myocarditis, but the anatomy is so specific. I mean, it's so weird to have these recurrent junctional rhythms. I mean, it's not strictly SVT, it's junctional. And is there anything about the endothelial supply and the blood supply to that node in your mind, as you think about it, that could explain, how the endotheliitis could explain the high junctional rhythms?
Absolutely. And that's, again, goes back to that CD147 receptor, the pericytes. So these little supporting cells that surround the smaller blood vessels, they're replete with that receptor. So wherever we find that receptor, and there was a good paper that came out just a week or two ago showing the preferential binding of S1 to that pericyte. Now we have pericytes in our capillaries in our brain as well. So I think there are two mechanisms there. One, it is concentrating the amount of spike in that pericyte, which allows it to transverse that wall into the surrounding tissues, yes. And number two, you also have portions of the brainstem responsible for rhythm in the sense of hearing. And so same thing, those pericytes in the brain, that CD147 receptor, that spike binding to those areas. Now you have two potential mechanisms of action for these arrhythmias and SVTs, et cetera, to be happening.
You have nanoparticles concentrating in the hypothalamus and in other areas that may well be affecting the rhythms. I know we're up against coming toward the end here. I have still 2 pages of things I want to talk with you about. So we'll have to make a date to do this again. Just so you know,
I think we should. By the way, I think you're absolutely correct.
I think so too. The way this show got started, Ryan, was really out of the repressive nature of this entire pandemic that Drew was seeing, and I certainly was a victim of it. This censorship, the ridicule, deridement, the fact that I was kicked off of every social media platform. And you are quite right that prior to this, in my 30 plus years as a practicing physician, robust, vigorous debate was a cornerstone of medicine. It's what we relied upon. And we would say the same thing. If you want to argue with me that I've misinterpreted the data, that I underestimated or overestimated the risk, that you've got a better study, bring it on. But let's have that discussion and let's really have it out. Respectful debate, but let's have that debate. I have been so appreciative of this platform and the ability for me to invite people like yourself and my colleagues of mine into this platform to have these open discussions that otherwise people really can't access. They sure as heck aren't getting it on the mainstream media, and they aren't getting it out there in their average Google search. So I appreciate you bringing this, your experience, your slides, your take on all of this to the table and being willing, as I said, fearless as you are, to bring it out there. And so thanks very much. We need to make a date for part 2, Ryan Cole part 2.
We do. Yeah. Yeah, we do. I agree. And
I just want to say thank you to you both, because it's not about ego. This is science on behalf of our fellow human beings, and that critical oath that we all took to, primum non nocere, first do no harm. And that's really why I've done what you guys have done, just trying to get information out to help people. And so I'm grateful for this opportunity.
And Dr. Cole, can people find more of your material at the rcolemd.com? Is that where like your slides and things are? People are on the restream are asking, where do I go? I want to see this again. So they can find that information there.
Yes.
Kelly, any other final thoughts?
No, I just,
go ahead.
Yeah.
I was going to say, I want to answer Dr. Drew's question about SVT, because the other thing too, I forgot to mention, that amyloid that we're seeing in vessels and some of those clots, that amyloid is also depositing between tissues in some organs in some autopsies. So that's a third mechanism. I wonder if that's
to keep an eye out on the literally the AV node, right? Because we're seeing these are not the usual SVTs. These are AV nodal, extremely rapid. that respond to ablation, but Jesus, you're ablating the AV. No, it's just a mess, and it's very specific. I've never seen this in young people before. It's usually people with advanced ischemic disease.
This is what's left of a 17-year-old. So if an autopsy is being done, and it should be done, I mean, my call to action for the medical profession is sudden deaths, make sure the autopsies are done, include the node. But if you're going to do an autopsy, then do the adequate markers for this protein that we can identify, do the adequate stains for amyloid. What a great question, because we know the mechanisms, both in the literature and under the microscope. So let's do it. Let's a call to action to our colleagues. Be willing to be the scientist again. Be willing to help your patients. Be they alive. Let the dead speak. Let's honor them with showing so others can learn and others cannot be harmed. So thank you guys so much. I super appreciate it.
Kelly, last words.
No, that's I was just going to say we know, and I've been reporting from the very, very beginning on the increases in cancers, increases in autoimmune diseases, increases in fertility issues, increase in cardiac issues. But what you are presenting here is the proof that these are related to these spike proteins and the spike proteins from the vaccines versus from native virus. And that's a very, very important thing. You are correct. You will not find what you don't look for. We are obligated to look for this, and we're obligated to report on what you're finding. So thanks again. And we will do this, as I said, for Dr. Ryan Cole, part 2. Date to be determined.
Thank you. Thank you, Dr. Victory. And we have some very great guests coming up. We have the Florida Surgeon General coming up, Dr. Lapido.
Surgeon General, yes.
Interesting, right?
Yes.
Yep.
Yes,
there
Dr.
he is,
Joe
Dr. Lapido.
Lapido coming next Wednesday, and then David Weissman, who Dr. Cole just referenced. We have Dr. Weissman joining us, and that date I think is the 14th of December. And then we're getting Dr. Bridle scheduled as well, Byron Bridle, folks who have worked closely with me and with Dr. Cole. So that's terrific.
And we will get Dr. Cole back on the schedule again for a part 2. It'll be particularly meaningful having already then interviewed Dr. Weissman and getting his input.
Yes.
And we'll probably have more questions after that. So that'd be very interesting. So Kelly, as always, thank you. I'm so glad to be back. Late to see you. And to everyone else out there, we appreciate you being here.
Thanks, Kelly.
We appreciate you on the restream.
Thanks.
We've been watching
Welcome
you the whole
back.
way. Thank you. Been trying to keep an eye on what you guys are saying.
Make sure you retweet Drew's tweet for Kelly's reinstatement.
Yes.
On
Oh,
Twitter.
please do, because the most irritating thing of in addition to the insult of having been banned for speaking truth, and I was really, the things I was kicked off for had nothing to do with my personal opinions. They were statements of fact, irrefutable fact, by the way. But the most irritating thing is that there is more than one sham, fake account out there. This particular one you're showing right now somehow got a verified blue check. This person is impersonating me, has stolen my
$8.
identity,
I don't think they're going to pay.
is taking credit for the work that I do for the different television shows I do, and for
it's
shows
not
like
you
this
speaking.
one.
So she's a snip. It might be even a guy. She's a tart. Okay, she's not. She doesn't even sound like you. She's not smart. She looks like an idiot. So I actually followed her. I didn't know.
It's illegal. It's
it is illegal.
masquerading
But a lot
as a
of people
physician. It's practicing medicine without a license. Very serious legal problem.
Yeah,
very serious.
Don't read the tweets. Just retweet. Let's get her reinstated and
Retweeted and
hopefully
please
we can get other doctors back on Twitter if they want to be on it or not. I would like you back.
Thank you.
We'll get you back.
I would.
I have
thank you. I appreciate
faith in
it.
Mr. Musk. All right, Kelly, thank you so much.
Thanks.
And I will be out the rest of the week. I've got to go to Austin.
I'm going to miss you.
I'm going to miss you too. And
2 days.
And when I know, it's weird. And when we come back, it's going to be the usual schedule, Tuesday, Wednesday, and Thursday. As we said on Wednesday, it'll be the Florida Surgeon General. I have a tremendous respect for that guy.
So excited about
I'm very excited about that one. So it'll be very interesting to talk to him.
Well, we had quite a showing today on Rumble, on YouTube, and all the other platforms. We appreciate your viewership, and we try to make as much information. Seems like the smarter the people are, the bigger the crowd is.
That's a good sign. I consider that a great sign. It gives me faith.
I couldn't understand half of the stuff that he said, but I kind of followed.
You can watch it again. It's not
doesn't
as though
sound good.
He was not using concepts that the average person could not understand. You may miss it because you're not familiar with it. Do watch it again. It's pretty easily understood. And Susan, I watched the Rumble rants very carefully today, and people
They were
were
really appropriate.
They were appropriate. They
We
were
didn't have
behaving
a-
themselves.
we didn't have
and
an
they were
imposter.
good. Very interesting comments too.
They didn't
So
say I had a stretched face or anything like that.
was that what they were saying before?
That's okay.
Oh my God.
I don't
right.
do anything.
Well, thank you guys.
It's the Genucell.
Thank you on the restream, and I will see you all next Tuesday. We'll see you then, 3:00 Pacific, as always. Ask Dr. Drew is produced by Caleb Nation and Susan Pinsky. As a reminder, the discussions here are not a substitute for medical care, diagnosis, or treatment. This show is intended for educational and informational purposes only. I am a licensed physician, but I am not a replacement for your personal doctor, and I am not practicing medicine here. Always remember that our understanding of medicine and science is constantly evolving. Though my opinion is based on the information that is available to me today, some of the contents of this show could be outdated in the future. Be sure to check with trusted resources in case any of the information has been updated since this was published. If you or someone you know is in immediate danger, don't call me, call 911. If you're feeling hopeless or suicidal, call the National Suicide Prevention Lifeline at 800-273-8255. You can find more of my recommended organizations and helpful resources at drdrew.com/help. My dad was a family practitioner, and he drilled into me, medicines are bad, bad, bad. It's only when you have to use them, you use them. I never had an antibiotic until I was like 17. And even then, he's like, all right, here we go. This is it. We're crossing a line here. You're going to take ampicillin. I'm like, okay.
See, I think your dad did it because he was smart. My dad did it because he was fucking cheap. You know what I mean? I think my dad did because I'm looking at it. He didn't fucking know anything. He was just like, hey, we're not buying nothing. We're not doing nothing.
These celebrities will step into our world. A world that's full of shock and pain.
I want to test the limits of who I am.
Why are you here?
10 years I was in a really abusive relationship. This is about taking back my own power.
Be afraid.
This is not an adventure race.
Be afraid.
No one's coming to save you. You will suffer together. That's the only way through.
Stand by me.
I have a child. Don't let me down.
All of you have what it takes physically to do this. Do you have what it takes mentally?
Do you still want to see if you're able to dig down deep?
What are you trying to get out of this?
Growing up, my sister became worldwide famous. I guess I just want to feel like I'm just worth something.
My idea of this course was about what I could get out of it and how it's all about what I can give.
If you should die, that's nature's way of saying you failed.
Oh, you got it coming!
That was rough.
Oh, stand by me. Stand by me.